Okay, let's have a little run through of DNA replication. Now, when our bodies are growing, replacing worn cells or repairing damaged tissues, the cells need to divide. And the first stage in this process is the replication of the DNA. Now, before we start talking about replication, let's just remind ourselves about the structure of a DNA molecule. Here we have a double helix shape held together with bases. And because of their shapes, A always bonds with T and C always bonds with G. The order of these bases tells the body the order of the amino acid sequence and therefore what kind of protein is going to be formed. So, for example, the code for hemoglobin protein is going to be different from the code from insulin or collagen. Now, don't be too worried about the complexity of this diagram here. It's not as tricky as it looks. So, at the very beginning, the DNA molecule has to be unzipped, just like a zipper on a jacket. And this is done by an enzyme called DNA helicase. And this leaves the bases exposed. Now this is all happening in the nucleus, and floating around in the nucleoplasm are lots of A, T, C and G bases. And with the help of something called DNA polymerase enzyme, these bases join in with the exposed ones. A's with T's and C's with G's, as I said. Now as the new molecule is made, they wind back up into a helix. And that's it. We have two new DNA molecules that are clones of the original. Now this process is known as semi-conservative, and this is because half of the new molecule already existed. So there's no need to manufacture a whole new DNA molecule from scratch because half of it was already there. Now at this point the cell is ready to continue with the process of cell division, and that pretty much is it. So here we are, a few questions. When does DNA replicate, and what are the stages of DNA replication? So if you just pause the video now, have a little go at answering those questions. Unpause it and I'll flick into the next slide with the answers on it. I hope that was useful.